Well, thank you very much, James, for such a truly kind introduction. I wish my mother's here to hear this introduction. I'd like to begin by thanking the CAS leadership for inviting me to speak at this very inspiring meeting. And I'm very excited as a stem cell researcher to share with you about my discoveries, and not only for scientific progress, but also as a part of my personal journey that reflects something much bigger, which is for each one of us here, as an immigrant from China, how our passion, perseverance, and dedication can bring to our contribution to this great country. I'd like to start then by talking about my personal journey. About 40-some years ago, when I graduated from high school on a small island in China, I never thought today I'd be standing here with all of you to share the excitement of science. And this little island actually is so small so that there's only one high school, and that's where I graduated. And this island actually is off the coast of Wenzhou City in China, which, as you know, is near Shanghai. And by saying near Shanghai, that's 300 miles away. So when I was a high school student, I actually was very interested in engineering, but not biology. Also at that time, China was experiencing a cultural revolution with no universities open. So my aspiration of being an engineer was to become an electrician after graduating from high school. And in the year when I graduated, the Cultural Revolution ended, and the colleges reopened their doors. And also around that time, the word genetic engineering became very trendy in China. So then I decided to become a genetic engineer, and for that reason, I joined the Fudan University to major in biochemistry, hoping that myself would become a member of this cool new breed of engineers. However, once I went to Fudan, I realized that biochemistry is not engineering. In fact, I feel biochemistry is even more exciting than engineering. So then I decided to pursue the biological side of study further. I went all the way to this side of the earth to Cornell University to study genetics and development. Consequently, China lost a potentially talented electrician. So at Cornell, I studied biological side and started to ask many questions. And by the time of graduation, I was fixed on stem cells. And in order to study stem cells, at that time was a very esoteric term. I looked around to find a lab that can accommodate my postdoc training. And I was very lucky that Dr. Alan Swelling's lab at the Carnegie Institution of Washington in Baltimore took me in. So after I started the stem cell project there, then I moved to Duke to start my own lab in the medical school. So at Duke, I enjoyed 12 fantastic years, and I thought I would just stay there for the rest of my career and then take a belated, I call the geriatric spring break to Florida. However, in 2006, something unexpected happened, which completely changed my career path. Yale called me to see whether I'd be interested as a candidate to establish a stem cell center at Yale. Well, a call from Yale, I better take it seriously. So once I visited Yale campus, I fell in love with this great university. And in fact, I was even in love with the city of New Haven, a very charming city. So in love so that I abandoned Florida as an American. Obviously, at Yale, we work on stem cells. So let me just talk to you about my research then. So how many of you heard about stem cells? Raise your hand. Wonderful, you guys are so scientifically savvy. So actually, there are two types of stem cells, as you probably all know. One type is called embryonic stem cells that exist in early embryos. Embryonic stem cells has the ability to produce all types of cells in our bodies, such as illustrated here, pancreatic cells, bones, and neurons. And there's a second type of stem cells, so-called adult tissue stem cells. As you can imagine, they only reside in our tissues. And normally they can only produce cell types within that tissue, but not outside that tissue. For example, neural stem cells can only produce neurons, but not bones or pancreatic cells. Despite the difference between these two major types of stem cells, they share a common ability. Namely, they all have the ability to self-renew. So-called, they can always make a copy of themselves. Therefore, they become immortal cells. And because of this immortality, quote unquote, these stem cells become basically the fountain of youth and the mother of all cells for our research. And since the discovery of stem cells in human about 30 or 40 years ago, now the entire new field really is blossomed. So when I entered the stem cell field, there were only very few people there, and I learned actually there are already two fundamental theories in this field. The first theory is called the stem cell asymmetric division theory, which is proposed about 150 years ago by a German scientist, Theodor Berger, in 1880s. At that time, he was smart enough to envision there must be some cells in our body, like the seeds of our life. And he imagined that stem cells can self-renew because they divide asymmetrically. For example, this stem cell, when it divides, you produce a daughter cell identical to yourself, and another daughter cell that becomes specialized, so-called commutative differentiation. That's an academic term. Now, this daughter stem cell, when it divides again, you will divide it asymmetrically again to produce another daughter stem cell and another type of differentiated daughter cells. So therefore, now after stem cell divides 3 times, as you can imagine, it can produce 3 different kinds of daughter cells, but yet still maintain itself there. So this asymmetric division later on became my career-long passion. Why stem cells have this amazing ability to self-renew. And when I was studying that, that led to the second theory in the field called the stem cell niche theory. So that theory was actually first proposed by British scientist Ray Richfield in 1978. When he realized that when you take blood cells from bone marrow, can always populate and regenerate much faster, say, than blood cells from spleen or periphery, then he proposed there must be some special locations in the bone marrow that are essential or at least promoting stem cells to self-renew. And so he called that the niche theory. So the stem cell niche theory, as I illustrate here, proposed that stem cells are hiding in an idyllic hideaway in a tissue, and that have this niche cells. And these niche cells will produce a signal range. When these stem cells are within this signal range, they happily self-renew and live forever. But once they leave this field, then they are doomed to differentiate and die young. So this was the stem cell niche theory. So how to prove this theory, which one existed for 140-some years, one also for a long time? I decided to tackle these 2 questions. However, it's not easy to test these 2 theories because of 2 issues specifically related to stem cells. The first issue is what I call the identity crisis. Normally, stem cells exist only in a very small number in our tissues, and they do not look different from any other differentiated cells that's right next to them. Therefore, even today, I have to be honest, many people claim they are studying stem cells, they still don't know exactly whether they're looking at stem cells. And so I thought that if we don't solve that problem, this will preclude any in-depth analysis on stem cell behavior and function. For example, you don't even know how they divide. Now the second theory, Dr. Williamson actually also mentioned there's a quantum physics concept called the Heisenberg's uncertainty principle. I feel that stem cells are the biological equivalent of these quantum particles in theoretical physics or nuclear physics. Namely, if you touch them, they change their form. This also precluded the analysis of stem cells. That's why the stem cell theory has been there for decades, over decades, however, very little progress is made. And how to overcome that problem? So I decided to choose a model stem cell system, which is better for me to resolve these issues. And I thought about a little fruit fly, Drosophila. How many of you have heard about little fruit fly? Wonderful, this is an amazing audience. So you know that little fruit flies, they have been over 100 years, a very powerful genetic model. Today, most of the genetic principles that we apply to human health are actually coming from the study of fruit flies. Then I decided if I use this little creature as a model, if they have stem cells, then I can quickly use the tools available for this little model to study stem cells. And I focus on little flies ovary because you see this ovarian unit is like a beads on a string structure. And if you see at this end here, there are cell division keep going on and they keep producing this long string of rectangles. So this gives me a sense there must be assembly like an organization and there must be stem cell at the tip. So in order to find the stem cells, I first took out this suspected region which possibly will have stem cells and transplant into another recipient flies. So I want to show how that structure, whether it can regenerate ovary. So my overall strategy was shown here. I want to first identify stem cells in fly ovary. If so, exactly where. And then after that, I want to study whether they divide asymmetrically, as people have been proposing almost 150 years. And after that, I want to know whether there's a niche existing for these stem cells. And of course, ultimate goal is to discover what genes are involved in regulating these stem cell divisions. So let's start with identifying stem cells. So I developed a method that allowed me to take out the tiny tip of the ovary and transplant it back into the abdomen of another recipient female fly whose ovary is being removed. And now you can see this result in real data. This is the tiny structure I transplanted, less than 100 micron. And then after that, we say in 12 hours, produced one egg chamber, 22 to 24 hours, another egg chambers, another 24 hours, we produce all these developing eggs. And eventually, within 3 days, it regenerated the entire ovary. So which told me that there must be stem cells there in that tiny structure. And excited by that, I even put that structure and put it into the tummy of male flies. And that can make male pregnant. Right. So I call these males Mr. Moms. So with this kind of success, I think they encouraged me to use non-laser microbeam to specifically kill specific cells. To see killing which cell will stop this process. And the reason is so simple, I don't need to explain to you. And turned out, only when I killed 2 anterior removed cells in this entire ovary, which is over a million cells, the entire process of egg production stops. If you kill any other cells, doesn't even slow down the process of egg production. Maybe just missing 1 or 2 members. So that clearly illustrates this organ actually has stem cells. And it's a stem cell driven process that leads to the production of eggs. And I also discovered right next to the stem cells is thin layer cells that I named as cap cells because they look like cap. So now we know where exactly the stem cells, we can test whether stem cells divide asymmetrically. And this is the real data I can show you. And turn out that they do divide asymmetrically to produce the daughter stem cell that still stay in contact with cap cell, and another daughter cell also differentiating and eventually become a mature egg. And the cap cells are also interesting because it's always staying there as a landmark. So to study whether stem cell is asymmetric, they divide or not, now you can see real data. So I'm circling this stem cell now under high magnification, and with the mitotic spindle, many of you probably know that ensuring the proper segregation DNA producer because it's going to win. And you have the cap cell right here, and as you can see that the stem cells indeed divide along the ovarian axis away from the cap cells. So that after each division, one daughter cell will still
remain in contact with cap cell, and we, by laser ablation, know that's still a stem cell. Whereas the other daughter cell, now we go away from the cap cell, and then become a non-stem cell. And we also discovered not only the cell fate is asymmetric, but even the cytological location and structure is asymmetric. We discovered a special that was specifically segregated and retained in the stem cell during the division. So that actually partly ensured important profile of stem cells. So knowing that stem cells indeed divide asymmetrically, we want to ask the next question: are there any nature cells there? And turned out we were suspicious of these cap cells and these cells because they're always right next to stem cells. In order to see whether these cells are indeed nature cells, we basically laser ablated these cap cells, and just partial ablation alone is sufficient to change stem cell dynamics. So we also using this simple model to prove actually there's a nature cells at this stage. Now, we are very happy because we did not stay in human and mammalian system. We went back to a much simpler system that allowed us to identify stem cells and to prove both important stem cell theories, the asymmetric division theory and the stem cell nature theory. So now the question really becomes, what genes control the asymmetric division of stem cells? Here I want to say using flies as a model has another advantage, because fly genome is only about 10% of human genome. So they can only afford to keep the top 10% most important genes in our humans. And the kind of genes we care about pathways or hair color, whatever, non-important. They cannot keep them, they keep it away. But the most important survival genes are in flies. So once you find the gene in fly, you know you find a blue chip equivalent gene in human. And you can then quickly go back to human to see whether that gene is there or not. And I bet you that gene will be there and not very similar. Just like DNA is genetic material, the DNA is everywhere, from bacteria all the way to human, in the same kind of structure. Now, we discovered that many stem cell genes, with a very small number of them, are important for the self-renewing, so-called immortality of stem cells. And as James kindly introduced, one family that we discovered was the Argonaut gene family, which encodes proteins with 4 functional regions. And if you do structural analysis, like quail-EM, just like our first speaker, Dr. Fung, beautifully have illustrated, you see a structure like this. So you can see that this structure, the PAG domain, the mid domain, they actually bind to RNAs. And according to the sequence and structural similarity, we subdivided this gene family or protein family into 2 subfamilies. One called the Argonaut and one called the PV subfamily. The Argonaut subfamily and PV subfamily share the common structure that can bind to small RNAs. And they also have a domain, just for experts here, it resembles the nucleus. So then we and others found that the Argonaut subfamily should bind to siRNAs and microRNAs. These RNAs are very short, non-coding, 21 nucleotides long, and they are ubiquitous in all cell types. And that's why today, when you see medical application, RNAi, it works in many cell types. And of course, these discoveries provide a basis for my other friends' great work on further studying those mechanisms. So as some of you may know, that the study of the siRNA leads to the discovery of RNA interference mechanism. So Craig Mello and Antifa got Nobel Prize in 2006. And last year, another Nobel Prize was given to the study of microRNAs by 2 other friends of mine, Victor Ambro and Gary Lofton, for this study of microRNAs. So what have I been studying in the meanwhile? I was interested more in the PV subfamily, because I saw Argonauts as housekeeping in every cell. But as a stem cell person, I thought it's more interesting for me to study this subfamily because it's only expressed in the germ cell, most primitive type of stem cells. And it turned out that we discovered, and soon confirmed by 3 other labs, this subfamily binds to yet another class of small RNAs. We named PV interacting RNAs, or now in the field people call PARNAs for short. And these RNAs are somewhat longer
than the microRNA and siRNAs, and they are only there in the productive cells and primitive stem cells. Now in order to study this RNA structure, let me briefly review with you the central dogma for molecular biology. This is the dogma that's guiding 97 or 98% of labs research in the biomedical field in the past 7 decades, which tells us that genetic information in DNA is transcribed into messenger RNAs, which are then further translated into proteins. And Dr. Frank's talk focused a lot on ribosomes, which actually is talking about how ribosomes make these messenger RNAs into proteins. And the discovery of microRNAs and siRNAs further enriched this dogma by saying that in addition to ribosomes, tRNAs, now there are 2 types of small RNA pathways involved in this process. However, when we discovered RNAs and look into where in the genome these RNAs are encoded, they are encoded in unlikely ways. So as you know, central dogma is important, but it really talks about only 1% of the genome that encodes protein. The vast majority of the genome are called junk DNA, it used to be called this way, and I prefer to call them terra incognita because really it's a field widely open we know nothing about. And to our nice surprise, the genes that encoding RNA are actually hiding in these junk DNA sequences. And we found that junk DNAs actually can encode many RNAs. In my lab, in all the major organisms, we've discovered at least 16 millions of different RNAs. Just to give you a comparison, our genome, the human genome, contains 23,000 genes. Now you suddenly, and there are only about 1,000 microRNAs. Suddenly there are 16 millions of RNAs, which is about 700 to 800 times greater than the total number of genes that's known to us. So we obviously are euphoric about these discoveries because if you liken our genome as a world, the traditional genes are like the old world. And suddenly we found ourselves landed ashore of a completely new world that encodes for different kinds of molecules. And so this is actually the pie I baked for my lab to celebrate the discovery of RNA. It is a pie RNA, as you can tell. And from this scoopy drawings, you can see I was so excited that my hands are trembling. So also this at that time was mentioned by scientists when it was 10 grade schools that James mentioned, even though we had no idea about what's the function. At the time it was just a phenomenon. In order to study the function of these RNAs, we decided to study its function genome because there are so many genes. So again, just briefly review, our human genome has 20,000 protein-coding genes that contains about 1% of the genome. And then there are vastly intergenic regions. Actually, there are many other genetic elements that exist that we know very little. We have about 1 million of transposons, so-called jumping genes. And jumping genes are often translated into RNAs in order to jump around, and they look like viruses. In fact, they're derived often from viruses. Therefore, people feel these are the invaders to our genome. We cannot do anything with them, just to copy. In addition to transposons, these are often called the selfish parasites in our genome. These few years, many labs, including my lab, discovered that 2 new types of major genes. One is encoding the long non-coding RNAs. These RNAs are longer, they don't have coding capacity. There are tens of thousands of that. And also the RNAs that we discovered, over millions of them. In addition, we should realize that our genome also contains a lot of pseudogenes, fake genes. These genes look like real genes, almost 100%, however, they are non-functional. They cannot make proteins. And in the current theory, these genes are viewed as this defunct carcass of active genes on their way out during evolution. And they can even make messenger RNAs beautiful. However, we should not forget that our genome also has structural components, centromeres that mediate DNA segregation, and telomeres that keep our genome intact. So despite the conspicuous presence of all these what I call the major genomic constituents, very little is known about their function or interregulatory relationship. So my lab's work in the past 15-ish years have shown that actually these genomic elements all have very active functions. For example, the RNAs can mediate transposon regulation of protein coding genes. In addition, pioneer can regulate the expression of pseudogenes and their regulation of the sister active genes. Furthermore, transposons can also regulate the expression of these linker RNAs through pioneers. And as I said, showing you all these are mediable pioneer pathways. And the latest we just found in the past few years that pioneer can even regulate the expression of centromere RNA. We always thought that if you read the textbook, even today, the centromere is the protein is the better part of DNA transcription RNA. No RNA is expressed, but actually they are expressing RNA, but they need to be controlled by pioneer. If you lose this control, the cell will not develop normal. And for stem cells, that's devastating. So just for non-scientists here, the meaning of this I can again metaphorically illustrate it in the next slide. If you liken our genome as a world, then in the old world now we know, in addition to the 23,000 traditional genes, there are also millions of transposons. And there are new world pioneer containing genes that's more than 16 million. And there are also tens of thousands of linker RNA genes. And we should not forget in our old world, there are also existence of pseudogenes. So I hope today I've shown you that these two worlds are tightly connected. Even centromeres and telomeres, what I showed you is pioneer genes can regulate the expression of traditional genes. Pioneer genes can regulate the expression of jumping genes. And pioneer can regulate the expression of linker RNAs. And even jumping genes can regulate the expression of traditional genes. And latest discovery showed that actually pseudogenes can regulate the expression of traditional genes, and pioneer can regulate even centromere and telomeres. So I think this kind of unified view, this united action, is especially important in today's real world. So you may think, well, you started your work with little flies, is that relevant in medical consequence? Yes. So we found out if these genes knock out stem cell quickly, if they become overactive, they lead to cancers. And we have shown in my lab that the gastric cancer, breast cancer, prostate cancer, testicular seminoma, and liver cancer, at least subtype of them, their occurrence is promoted and requires the overexpression of these PE genes that we have found. And even our latest result for colorectal cancer. So my dream is hopefully this will give us a third generation of so-called cancer therapy that's more advanced even and complementary to the current second generation called the immunotherapy-based therapy. And we really are working on this pretty hard. And to end my talk, I'd like to acknowledge generations of trainees in my lab, really who did a lot of work, therefore I can do lots of talking here. Starting with my Duke trainees, I'm very glad my Duke former colleagues also here, and also my first generation of trainee at Yale, the second generation, the third generation, wonderful people, very, very successful. So I'd like to stop here, and thank you very much for your attention.